Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us, the 132nd seminar, and I call it on Antheia Day. Yesterday, I came back to my St. Louis home after my 3 weeks trips to San Diego, Iceland, and Boston to move to San Diego and then attend multiple conferences. Nothing is more important than family and also health, I believe. Although I enjoyed meeting many of my friends, colleagues, and also the best meal is, of course, anything we eat with beloved people. Actually, I enjoyed my dinner yesterday with my family. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Christina Smolke. She doesn't need any introduction because we all know her. She is now the CEO of Antheia, a biotechnology company. She used to be a professor at Caltech and Stanford University with a very active and productive career with many big journal papers. I admire her, papers all the time and her work. She received her PhD degree from UC Berkeley under the guidance of Professor J. Kisling, and thus she is my also academic aunt. Her passion for innovation and technology translation made her start her biotechnology company, and I hope her company will deliver many important drugs to the market. Unfortunately, she is currently feeling a little bit sick today, so she couldn't, basically, show her right now. So basically, her video is off today, but I'm glad she made it today. Christina, thank you so much for your amazing contribution to the scientific community and also the biotech industry, and the virtual podium is all yours now. And thank you again.
Thank you so much. I'm going to pull up my slides and get started with my brief comments. So I thought I'd just give a very quick overview of what we've been working on, both at Antheia and how it leverages some of the original scientific breakthroughs that we had developed in my labs at Stanford and Caltech. So the focus is really on rebuilding critical supply chains with synthetic biology. And to start with, a lot of the work we're doing now at Antheia is leveraging the pioneering work that we had done over about 15 to 20 years of academic research that was done at Stanford and Caltech, where we really pushed the boundaries of the types of molecules that you can synthesize with biosynthesis and fermentation. And really, I think what was notable and groundbreaking about this work was the complexity of biosynthetic pathways that we were able to engineer within microorganisms like yeast, with a focus on compounds that had bioactivities and actually served as active pharmaceutical ingredients for critical medicines in our supply chains. But ultimately, what we highlight here is just some of the early discoveries and scientific breakthroughs that demonstrated really the complexity of biosynthetic pathways that we could build into yeast. And then focusing on where we push these discoveries and the problems that we're working to solve, it's really one of equitable access to essential medicines. So this is a really, it is an urgent and pressing need. Even in the United States, drug shortages are continuing to increase in frequency and duration. But even if we look broadly across the globe, there's 2 billion people that lack access to basic medicines. And I'm not talking about the latest, cell therapy or gene therapy to come onto the market, that cost millions of dollars per dose. I'm talking about essential medicines like, children's cold medicine or chemotherapeutics or ADHD medicines, things that are really just critical to public health that are increasingly in shortage. And when you look at some of the reasons that are contributing to this problem, it's certainly multifaceted, but one of them is just the way that our global supply chains have evolved and the vulnerabilities and fragilities that exist within those supply chains, specifically our reliance on very few geographical regions to source these and the susceptibility to single point failures in the way the supply chains have evolved. And in particular, a very significant fraction of our medicines actually rely on agricultural sourcing. And that's because they're sufficiently complex, the molecules are, those chemicals, that we can't synthesize them efficiently with synthetic chemistry. So we started Antheia with the mission to be able to leverage next generation synthetic biology, the type of science that we had developed in the labs at Stanford and Caltech, to be able to really transform pharmaceutical supply chains, to bring manufacturing and innovation to the industry, to allow these supply chains to be more efficient, more resistant to failures, more resilient, and also more agile, ultimately to be able to address issues of access to medicine and affordability broadly. So if we take a step into the challenges, you think about just manufacturing principles when you're reliant on agriculture, sort of growing medicinal plants or a marine sponge or an animal to basically harvest a pharmaceutical ingredient from that source. A manufacturing cycle time can take over 100 weeks, and that's a very long time to complete that manufacturing cycle time. So we began to look at what if you could leverage a different type of production host, and in fact, brewer's yeast, where you can transform your manufacturing cycle time by 50x reduction to be less than 2 weeks. Ultimately, then, what we're able to do is leverage this whole cell engineering technique that we developed in our laboratories, initially, again, in academia, where we turn yeast into a miniature drug factory, able to take a simple starting molecule like sugar, and then in a one-step fermentation process. And I think that's really important from a process perspective, that you're doing all the synthesis in one manufacturing step to produce that complex pharmaceutical ingredient. Now, of course, the reality is much more complex than that schematic cartoon I showed you in the last slide. And this is an example of one of those drug factories that we had designed in the laboratory most recently for a class of compounds that make essential medicines like scopolamine and atropine. But what you can see here is that, in fact, it's very complicated in the number of biochemical steps that you are actually coordinating within the cell, and the number of enzymes that you have to basically get to work together to do all of these synthesis steps within the cell directly. One of the things that we found was very powerful in terms of advancing the research was the intersection of bringing computational techniques, machine learning, with genomics and the biology to really broaden this platform. And so I'm highlighting here some of the work that we've done that shows the application of different computational techniques, both standard techniques like different types of linear regression analysis on genome data to identify new chemistries, applying neural network models on genomic data to identify new classes of proteins that play a role in these biosynthetic pathways, as well as computational tools and databases to search and expand chemistries to develop new to nature molecules. All of these become really important when we think about broadening the platform to additional chemistries and molecules over time. And then the final thing I want to share is just that this science is now making medicines at scale. We are producing our first product via a validated biomanufacturing process at 120,000 liter fermentation scale now in our European manufacturing partner. So this is a situation and an example where, we've had success in taking breakthrough science that was developed, in the laboratories and now moving it into a commercial product. And this product is being used by our customers to make about half a dozen different critical medicines. I just want to highlight this is the first time that this compound has been made outside of the native plant. So the first time that a biosynthetic version of this compound is actually being brought to the market. So that's a huge milestone. And now we're set up to be bringing multiple pharmaceutical ingredients to the market over the next several years, being able to really leverage that engine and the platform that I described in the previous slide. So I think this is a really exciting example of just science making its way into actual, impact in an industry. And the final thing I'll just end with is highlighting the advantages of these types of approaches, both in terms of time, manufacturing time, right? Being able to reduce that by about 50x from 100 weeks to 2 weeks. Cost is important, being able to decrease cost from either 10x or even higher. And then finally, from a sustainability perspective, there's a huge decrease in the amount of arable land that's required. When you look at the sugar that's required to grow the yeast versus the land that's required to grow the crops and animals, about 10,000x reduction in renewable resources. So I think all of this is really significant when we think about what synthetic biology can bring to the industry. So I'll just end there, say thank you, and appreciate the time to be able to give an update on what we're doing here today.
Oh, thank you so much. This is amazing. So, I believe this short talk basically showing your entire journey within very short time. Not only technical content, but at the same time, all the difficulty you probably already faced last several years or even 2 decades. You gave amazing talk as usual, and also even though you are not feeling good today. Also, one connection I found today, I absolutely thinks that interesting. Your factory is now in Italy. That actually I believe the Simone's hometown, I believe, and that's an interesting connection I have today. So thank you so much again, and I wish you recover very soon.
Thank you.
Yeah, thank you. All right, so now the main speaker of today with a longer introduction, Dr. Simone Giaveri, originally from Italy, as I mentioned, is a materials scientist working at the interface between bio-inspired materials, cell-free synthetic biology, and bottom-up synthetic metabolism. He received his MS degree in material engineering and nanotechnology, cum laude, at Politecnico di Milano in 2015. In 2016, he joined the Material Science and Engineering Department at the École Polytechnique Fédérale de Lausanne, in short EPFL, Switzerland, to carry out his PhD program at the interface between material science and synthetic biology. In his doctoral studies, he contributed to the early development of a new vision about recycling plastics inspired by nature. This study demonstrated as a proof of concept that it is possible to envision a circular economy model for polymers where materials are transformed into new materials, not necessarily related to the parent one. After defending his thesis in 2021, he joined the Department of Biochemistry and Synthetic Metabolism at the Max Planck Institute for Terrestrial Microbiology in Marburg, Germany, where he is currently working with Professor Tobias Erb to develop a self-regenerating metabolic and genetic linked in vitro network. In fact, I visited this lab to give a seminar I think last year, and I also met Simone at the conference this year. For this work, he was awarded the EMBO postdoctoral fellowship in 2022. I think he's a really truly a rising star with a big potential, and then recently published big paper. I believe he will talk about that work today. Simone, thanks so much for your time today, and please take it away. It's all yours now. Thank you.
Okay, let me share my presentation. Okay, so thank you very much, Moon, for giving me the opportunity to speak about the research of my last years here at the SynBYSS seminar series. Thank you very much for your kind introduction. And I hope you can all hear me well and see well my screen.
Yes.
Great. Okay, I also would like quickly to thank Christina Smolke for joining us before as a pioneer speaker in the field. Thank you very much, Christina, for giving like a short but very inspiring introduction. So today in this talk, I would like mainly to talk about the postdoctoral work I have carried out at the Max Planck Institute for Terrestrial Microbiology in Marburg, in Germany, in the department of Tobias Erb. But in reality, at the end, I decided to connect this work with my previous PhD research at EPFL in Lausanne, Switzerland, to give a little bit more broad overview about my research in the last years. And then in the end, I actually would like to conclude this presentation with a few slides, a few ideas that are related to my future research to show what are my current interests and current passion in research to become independent group leader. So I would like now to quickly start this presentation with a brief introduction about my path in science that has all have been at the interface between cell-free synthetic biology and material science. And this because of the global audience of this seminar, because at least people that are joining us today from all over the world can see where the research that I'm going to talk about during the seminar has been carried out in the different European countries. And as you can see here from the screen, from the red dot in the screen, I started a long time ago from Milano, from Polytechnic di Milano, where I studied materials engineering and nanotechnology. And then I moved up to EPFL in Lausanne, Switzerland, for a PhD also in material science. But in reality, it was really at the interface between material science and cell-free synthetic biology. And for this reason, I was advised by Francesco Stellacci for the material science point of view and from Sebastian Merkel for the cell-free synthetic biology part of the PhD. And I really have to thank Sebastian for introducing me to the field of synthetic biology, because for me it became very fascinating field. I thought about a cool idea I would like to work on as an independent group leader in the future. And so I decided that the best option to try to carry out this project in the future as independent group leader was to join the department of Tobias Erb at the Max Planck Institute in Marburg, where I am currently, to work on cell-free synthetic metabolism, as Toby is one of the world leaders in this field. And I really have to thank Toby for all his support, as well as the Max Planck Society for all the resources and equipment that I could use for my research here at the Institute. And finally, I also would like to thank EMBO for funding my postdoc here at the MPI Marburg. So after this very long introduction, let's start talking about science finally. And as you all know pretty well, a lot of work has been carried out in the last years in the field of bottom-up synthetic biology in order to assemble the so-called minimal or artificial cells from non-living components, or better say, to attempt to perform this, to assemble minimal cells. And this research has been all carried out in the field of compartmentalization, communication between different compartments, energy supply, and metabolism. Here I would like already to highlight the cat cycle, because we are going to talk a lot about it later on in the presentation. And then DNA replication, protein production. Again, here I would like to highlight the pure system, as well as growth and division and motility. But as a material scientist, there is a property typical of living systems that I particularly admire, and this is their ability to self-regenerate and repair autonomously. And I think that this regard, a keynote work has been performed by Barbora Lavikova and Nathanaela Kunakorn in the Merkel lab at the EPFL a few years ago, back in 2020, when I was there also as a PhD student. And what they did is that they were able to pioneer self-regeneration outside cells by using transcription and translation in microfluidic reactors. And more in detail, what they did is that they were able to assemble a pure system that was able to self-regenerate once it was provided with the instruction encoded into the DNA by using the anabolic tool of transcription and translation, and once the amino acid building blocks were provided to the system. And this work for me was really like great source of inspiration for my research in the last years. And I think that, but I think that in nature, what happens is that living cells are actually open systems and are able to make use of energy sources as well as matter in the form of building blocks or precursors from the environment to autonomously synthesize the molecules, the compounds, as well as the macromolecules of need. And all of this is thanks to metabolism, is because of metabolism that will become the key word of this presentation, if you allow me, and it is the dynamic network of all chemical reaction happening in a cell that provides living systems with the energy and the building blocks that are necessary for life. And so the idea when I joined the department of Tobias Herb here at the Max Planck Institute in Marburg was to see if I could bottom-up assemble a metabolic and genetic linked in vitro network that I shorten as MGLN, that is able again to use the information encoded into the DNA as well as the anabolic tool of transcription and translation in the pure system. But this time it is able to autonomously synthesize, at least partially, the needed building blocks for regeneration by its own metabolism. And in our case, this means amino acid and also means from CO2, that is inorganic carbon. So how to do this in practice? Well, the idea was to start from the crotonyl coenzyme A, ethylmalonyl coenzyme A, hydroxybutyryl coenzyme A, cycle that we normally shorten as CATCH, that is a cell-free, it means it works outside cells. It is a synthetic, it means it does not exist in nature, and is a CO2 fixing system that is able to start from the C3 molecule propional-CoA, fix 2 CO2 molecules per turn, and produce glyoxylate, as well as regenerate propional-CoA. And this cycle was invented a few years ago by Tobias Herb. And the idea was to integrate this cycle together with a special pure system that does contain all the necessary elements for transcription and translation apart from the amino acid glycine. And the idea was to convert the glyoxylate produced from CO2 into glycine by using an aminotransferase enzyme. And then, so this glycine would activate the translation in the pure system. So all this looks like very pretty, I think, on the paper, on the slide, but how actually to do this in practice in the lab? So the first step was actually to try to assemble a so-called delta glycine pure system. That is a pure system that depends on the externally added glycine in order to perform translation. And in fact, if you see for the transcription translation of a EGFP gene that is sketched here, you can see that if there is no glycine in the system, then there is no fluorescent signal recorded by the plate reader. As soon as like a 300 micromolar glycine is given to the system, then you obtain a very nice fluorescent plateau. And so I was also very interested to see if I could identify a range of concentration of glycine for which I could find a linear dependence between glycine concentration and fluorescent plateau achieved. And you can see here in the slide that every time I was doubling the concentration of glycine, I was obtaining a 2 times higher fluorescent plateau in the plate reader in the range between 10 micromolar and 150 micromolar. And I was really happy about this experiment because it meant that I could have control really on the system. And then this was the moment of combining together these results with the choice of the key enzyme to link the genetic layer of the pure with the metabolic layer of the catch. That is the choice of the key enzyme. This is the aminotransferase for this reaction to happen between glyoxylate and glycine. And the choice of this enzyme is very important because this reaction should happen in the presence of all of the amino acid residues, and you don't want to produce glycine out of another residue, but you want to produce glycine out of CO2. And so after a few tests, what we did is that we were able to identify the glutamate aminotransferase GT1 from Arabidopsis thaliana. And why did we choose this enzyme? Because it was able to produce more than 200 micromolar of glycine after 15 minutes without modifying the concentration of any other amino acid residue in the pool. And so, as I was saying before, this was the time actually to combine together this enzyme with the delta glycine pure system that now, if you want, we can call delta glyoxylate pure system. And I basically repeated the same experiment I commented before, substituting glycine with glyoxylate and keeping constant the concentration of the identified GT1 aminotransferase. And you can see again, in this case, that in the interval of range between 10 micromolar of glyoxylate and 150 micromolar of glyoxylate, I was obtaining a linear dependence between amount of glyoxylate and fluorescent plateau achieved in the plate reader. So I was really happy about this experiment. This was the time to start combining together the two layers. And I decided to do this initially by keeping off the metabolic layer of the catch. And this I did without providing propionyl-CoA to the cat cycle, and switch on the genetic layer of the pure system by providing a DNA template to the system encoding for EGFP and providing glycine. And what I could see is that once the cat cycle was diluted 5 times into the pure system, then the pure system could produce a signal for the sample curve in the plate reader for the transcription and translation of GFP once it was supplemented with 300 micromolar glycine. It was more than 1 order of magnitude higher with respect to negative control. And so I could have a starting point to start to play with. And then what I did exactly the opposite. So I decided to now switch on the metabolic layer of the cat cycle by providing propionyl-CoA to the system and keep off the pure system. That means I did not provide any DNA sequence to the overall system. And in this case, I was interested to investigate how much glycine could a 5 times diluted cat cycle produce in the presence of all the components of the pure system. And after a few trials, I was very impressed to see that we could already produce about 110 micromolar glycine, as I said, having the cat cycle diluted 5 times and in the presence of all the components of the pure system. And this was really nice also because this 110 micromolar is inside this linear range I was mentioning before between amount of glycine and fluorescent plateau that I could achieve in the pure. So actually it was already pretty good starting point. But at the same time, I was also very curious to see if I could optimize this result to make this system better. And here I have to really thank Nitin Bora, that was the master student in our department that I was supervising a couple of years ago, and that now is a PhD student in our department, and that helped me actually optimizing the system by adding one by one the components of the pure system on top of the cat cycle diluted and measure how much glycine we're obtaining. And you can see now that after some optimization, we were able to improve the system about 3 times, so producing about 300 micromolar glycine in the end. And this was really cool. And so this was the time not only to combine the two systems together, but really to integrate these two systems of the catch and the pure together. And you can see now that if both layers are active, that means the metabolic layer is fed with propionyl-CoA, the GT1 aminotransferase is present into the system, and the pure is also active because it is fed with the DNA template encoding for EGFP, then you can see a very nice signal into the plate reader because the CO2 gets fixed into glyoxylate that gets converted into glycine, and glycine is used for transcription and translation. And this means that finally the MGLN, that is the metabolic and genetic linked in vitro network, is finally set. And I was really happy about this, of course, about this result. But now if I look a little bit from farther away this system, what I notice is that up to now, at least in our MGLN, what happens is that there is a link between the metabolism of the catch and the genetic machinery of the pure system. But this link actually works only in one direction, because metabolism provides a building block for the genetic machinery, but there is no way back. But on the contrary, what happens in living systems is that living systems are able to self-regenerate and self-sustain through cyclical processes, where the genetic machinery encodes for enzymes working in the metabolic layer, and the enzymes in metabolism provides the genetic machinery with building blocks such as amino acids as well as nucleotides that are necessary for the genetic machinery to process the genetic informations. And so I really wanted to try to mimic this property typical of living systems in our MGLN. And then how to do this actually. And so to do this, I tried to make these two layers of metabolism and genetics actually interdependent with one another. And to do this, I started by setting up a self-integrating MGLN that is composed, where you can see like all the components are present in the cat cycle, in the metabolism, all the components are present in the pure system, apart from glycine, obviously. But there is the key enzyme, the link between the two systems, that is the GT1 aminotransferase, that is actually missing. And so initially I decided to switch on the metabolism of the catch cycle that you can see here. It's turning, producing glyoxylate, but keeping off the pure layer. And in this case, of course, I could not measure any fluorescent plateau for transcription and translation in the plate reader because the pure is inactive, there is no DNA. I then decided to switch off the cat cycle and switch on the pure system by providing 2 DNA templates to MGLN. One DNA template encodes for a GFP reporter, and the second DNA template encodes for the missing GT1 aminotransferase that links the two systems. But again, I could not record any fluorescence. And why? Because obviously glycine is missing in the pure system, and so translation couldn't happen. And then I decided actually now to activate the MGLN by providing the system with minimal concentration of glycine, about 10 micromolar. And this concentration of glycine was enough to produce minimal concentrations of the GT1 missing enzyme. You can see here it appeared in the sketch, as well as also minimal concentrations of GFP. But what you can also see is that after about 30-40 minutes, the system stopped working. We got to plateau for GFP production, and means it went to equilibrium. And why? Because glycine was already consumed because it was present in very low amount. And this was the time I actually tried to push the system out of equilibrium by switching on metabolism. And so I provided propanol-CoA to the cat cycle that now could start turning, fixing CO2 into glyoxylate that could be turned now into glycine because of the small amount of GT1 previously produced by the genetic layer of the pure. And this glycine was used to produce actually more GT1 enzyme, and this in an autocatalytic fashion. And you can see here from the plate reader that now the system could work for about 12 hours. So we went from a working time of about 30-40 minutes until almost 12 hours. So this was, of course, very interesting. And it was also the time now to finally work on self-regeneration. That was the initial driving force from the great work I mentioned before that was carried out in the Merkel lab at EPFL. And to do this, I decided to assemble a very special MGLN where actually the metabolic layer of the catch misses two enzymes that are key for CO2 fixation. And these enzymes are the ethyl malonyl-CoA mutase that we normally shorten as ECM, and the methyl ethyl malonyl-CoA epimerase that we shorten as AP, that you can see here are missing in these two steps of the cat cycle. And so what happens is that now when I switch on the metabolic layer of the catch and keep off the pure, now propanol-CoA can be converted until methyl malonyl-CoA, but methyl malonyl-CoA cannot be converted into succinyl-CoA. And this is because the AP enzyme is missing in the metabolic cycle. And then I proceed to work in the same fashion as I did before. That is, I switched off the metabolism and switch on the genetic layer of the pure system, this time by providing the MGLN with the 3 DNA templates encoding for, again, EGFP to have like a fluorescent signal interpreter reader, as well as the 2 DNA sequences encoding for the 2 missing enzymes of the metabolic layer of the catch. And you can see again that there is no signal interpret reader, and this is because obviously glycine is missing. As I did before, I provided MGLN with 10 micromolar glycine, and this glycine was used immediately to produce the missing enzyme in the cat cycle. You can see that now appearing them here in the sketch, as well as minimal concentrations of GFP, as you can see from the plate reader plot. But again, the system went to equilibrium quite fast after about 30 minutes. And again, I decided to switch on now the metabolism to push the MGLN out of equilibrium. And now, actually, methyl malonyl-CoA could be converted into succinyl-CoA because AP was produced by the pure system, and same with ECM. And so the catch could start turning, producing glyoxylate out of CO2, and this glyoxylate could be converted into glycine. And this glycine could be used to produce, again, more enzymes for the metabolism. And this, again, in a feed-forward fashion, meaning that I was able to assemble outside cells MGLN, so a cell-free system whose activity of its metabolic layer of the catch could be improved by the activity of the genetic layer of the pure. And I was really like fascinated by this result. And I was even actually more happy when I was able to run this whole system that is composed by 53 enzymes coming from all domains of life inside cell-size droplets. And so actually up to now, what we did is that we spoke about metabolism to provide building blocks, so in this case, amino acids. But as I mentioned at the beginning of this talk, metabolism does not only provide building blocks, but can also provide energy to living systems. In this regard, I think that a keynote work has been performed a few years ago in our department by Terr and Miller that was able to power the cat cycle with light by using thylakoid membranes that were able to regenerate the ATP and the NADPH necessary for the cat to turn. And so, based on this work, what I'm currently trying to do with Hao Yuan, that is a master student in the department, is to set up and to use this system to power a special pure that is named that we named Delta E pure. That is a pure that lacks the energy regeneration module and can regenerate energy ATP from light. And this work is in process, let's say, together with Hao Yuan, that was a master student in the department that I supervised and now just started his PhD at the Technical University of Munich. But energy does not necessarily mean light, can also mean electricity. And then I was more than happy to collaborate with my colleague, Dr. Luo, a couple of years ago, also here in the department, in the herb department, who was able, Shan to set up a new to nature electrobiological cycle module that is able to produce ATP out of electricity with a minimal sets of biological components and without any membrane. And for this reason, actually, it is ideal, this system, to be coupled with and integrated with in vitro systems. And in fact, I was in charge of actually integrate this system with the transcription and translation in the Delta E pure that I mentioned before. And you can see immediately from the plot that once the key enzyme of the AAA cycle, that is the aldehyde oxidoreductase enzyme, is present, then the cycle can turn and produce ATP, and I could use this ATP for synthesizing mRNA in the pure system and then to power translation as well. Instead, once the aldehyde oxidoreductase enzyme was not present in the system, then the cycle could not turn, and so I could not have ATP to perform transcription and translation. But as I mentioned also before at the beginning of the presentation, metabolism is really like a dynamic network of chemical reactions that continuously replenish metabolic intermediates that are consumed during the biosynthetic routes. And a great work in this regard, in my opinion, has been carried out in our department a couple of years ago also by Dr. Deal, that was able to integrate, better say, the cat cycle with synthetic anaplerotic modules that were able to regenerate, to replenish, let's better say, some core intermediates of the cat cycle that were drained away by synthesizing polyketide. And then what I'm currently doing with Nitin Bora is to develop another MGLN that is able to switch on its genetic layer upon reaching a certain concentration of a metabolite that is glycolate produced by the cat cycle. And so once this concentration of glycolate has been reached, this MGLN is able to autonomously activate an anaplerotic cycle that could feed back intermediates to the cat cycle, allowing the cat cycle to run for a longer time, so somehow to repair the system. But again, up to now, I spoke about metabolism mainly for biosynthesis. But in reality, what happens is that living cells under special circumstances of stress, such as, for example, nutrient starvation, are able to recycle macromolecules such as proteins or nucleic acids in a process that is named autophagy. And then during my PhD in the Stellacci and Merkel labs at the EPFL, I was actually able to show that peptides, as well as mixtures of peptides, as well as mixtures of peptides and engineering relevant proteins such as silk or beta-lactoglobulin could be recycled outside cells without the need of any separation of the initial material. And this was also possible in multiple cycles, every time producing a different material and the material of need. And in addition to this is not only limited to proteins, but I was able to collaborate with Dr. Liu, also from the Stellacci lab, who was able to recycle calf DNA into monophosphates, nucleotide monophosphates, and then after phosphorylation could be used again to synthesize other DNA sequences that I was able to use for transcription and translation in the pure. And so you have seen today now in my long presentation that we integrated together many different components coming from all domains of life in the different works that I worked on and described today. But if we want in the future to build autonomous systems, that is systems able to use the energy and matter from the environment in order to perform their functions, then we will need even to integrate and combine more components coming from all domains of life. And so in this case, what is important is to take advantage of synergies, but also trying to limit negative interactions between components that are always present. So how do we do this at the moment? So at the moment, we use active learning workflows coupled with robotic platforms. And you can see here, for example, one example of an approach that we are currently using in the lab that was invented a couple of years ago by Dr. Pandi, also in our department. And currently, I am applying this together with Nitin Bora to the decision-making MGLN I just introduced before in order to optimize this system. And we just started to use this approach to our system. And you can see that just after only few rounds, only 3 rounds of optimization, we are already starting slowly to increase the performances of the system. This is all at the beginning, so let's see how it will end up. So, finally, every time you work with the metabolic cycles, you know that you need prototyping. And this means actually injecting thousands of samples into the mass spectrometer. And then I can say that in our institute, at the Max Planck Institute, we have a lot of expertise, a lot of resources, and a lot of also machines, of course. But it's also true that this takes quite a lot of time. And so I was more than happy to collaborate with my colleague, Sebastian Bartel, that developed an in vitro-based, transcription-based sensor that is fast, allows high throughput, it does not require long sample preparation, and it also, let's say, not expensive, but also not expensive also in terms of also time and working time. And this is like, I was more than happy to test, to help Sebastian to test his sensor on different catch cycles in order to use this strategy as a complementary and alternative strategy with respect to mass spectrometry. And then now I actually would like to conclude this long talk by quickly talk about what I'm really fascinated about in research at the moment, especially for my let's say the research plant as an independent group leader in the future. And then as most of you know very well, a lot of work has been performed in material science in order to assemble materials with lifelike properties, and this by integrating together, by combining together engineered cells with non-living matrices in the so-called engineered living materials that we normally shorten as ELMs. And these materials has been used for a plethora of different applications, ranging from structural applications, for example, biobricks, bioglues, but even to medical and biomedical applications. And here in the slide is one of the very recent last papers like on this in this field that I really liked. And then on the same, like, let's say in parallel, but with a different approach, that is by using the bottom-up approach that we discussed for long in my presentation today, synthetic biologists in the last years were able to apply, to use for the first time cell mimics for medical applications, for example, in the field of tissue angiogenesis, as well as in neural differentiation. And so at the interface between these two fields, what I would like to do as an independent group leader is to open a research line for developing metabolically active materials that are soft materials that are equipped with cell mimics that are loaded with cell-free metabolic reactions, and with this they are able to interact with the living cells for therapeutic applications. And how do they work? They should work in a sense, compute, and react fashion, and they should be able to take advantage of resources, that means energy and matter, from the environment where they work, and use these resources through metabolism to produce an output molecule of need. And really like using metabolism really like an engine. And I'm currently actually looking for the best place in the world to start this research line. And with this, I would like really to conclude this presentation by thanking again a lot Tobias Erb for all his support. I would like to thank you very much the students I supervise that are Nitin Bora, now PhD student in the department, Ha Yuan for his master thesis, then all the collaborators that are listed there. I really would like to thank also my PhD supervisors from EPFL, Francesco Stellacci and Sebastian Merkel. And I would like to thank EMBO again for funding my research and the Max Planck. And thank you very much all for your attention. And I'm here happy to take any question.
Amazing talk. I really like it. So let me see. I see the question from audience already.
Yes.
Two questions I got. The first question from Canyon, Alexander, is this work repurposing the CET-CH cycle? I thought it was built as a carbon fixing alternative to the Kelvin cycle rather than as a glycine production pathway.
Well, I would say it's a different story, I would say. So what so let's say as a material scientist, so this is like the reason in reality why I decided to join the Arabic department. So I have no, let's say, super expertise about metabolism from my PhD, and so I didn't, let's say, discover a new metabolic cycle, a new metabolic pathway, but I wanted to make use of the CAT cycle that was invented by Tobias Erb to produce something and that I needed, for example, in this specific case of my postdoc, to build this interdependent network where metabolism provides building blocks for the genetics to work, and the genetic in turn encode for enzymes of the metabolic layer. Let's say I would say the answer is neither yes or no. So I didn't want to repurpose the catch. the catch is there for, of course, for CO2 fixation, but I wanted to make use of the catch in order to build a system, a cell-free system, where both genetic layer and metabolic layer could interact, and all of each other in a back and forth manner, as I showed in the presentation, and this like trying to mimic these interdependent connections that are there in the living systems. I hope I answered detailed enough.
Okay, the second question by the same person.
Yes.
Is the active learning process an example of directed evolution?
No. So let's say how it works with this active learning process. So active learning is working like a machine learning. So what happens is that initially, for the first time, the algorithm, let's say, suggests you some experiments in terms of concentrations or amounts, depending on how you build it, but like of components to use, and then that is basically almost random. And then what happens is that you run these experiments, and then you take the results of this experiment and you feed these results back into the algorithm. And then after a few cycles, then what happens is that the algorithm is able to suggest you combinations of components that are working hopefully better and better. And this is not possible to do it by hand because there are too many components in the system. Specifically in the MGLN, there are more than 100 components, 53 enzymes, only enzymes. So I don't think it would be really possible by hand to explore all the possible optimizations and combinations, and there would be definitely too much. So this is the way it works.
Okay. I don't see any more question. I will ask one question. Out of my curiosity.
You're welcome to.
So, I fascinate by your presentation, and then I keep thinking about future.
Yes.
Whatever, everything just done beautifully and perfectly. I now imagine one day or someday,
Yes.
Those researcher successfully make membrane-like materials, and then in that material inside there are all components you just described, including recyclability you just mentioned.
Yes.
And then that is dream of all the synthetic cell community people, I believe.
Yes.
And then let's say that happened.
Yes.
And then let's say you able to make recycle possible.
Yes.
And then now I am JCBI, and then I also try to think about from the synthetic cell perspective. But
Yes.
JCBI synthetic cell is something different because we use the cell originally, coming from real cell, but get rid of the genome and then transplant the entire synthetic genome and then the reboot. That's the first demonstration, but now they try to do something different. Well, question is to approach completely bottom up, semi bottom up. Will they same? Will they be same in terms of function? And also second question is I start to do some aging project.
Yeah.
If you perfectly recycle everything, you will make cell never die. That's my second question. It's kind of more philosophical question, I believe, but I want to ask.
I start with the second question and then trying to. They're all good, like very good questions. So that's the point. So on what side, what happens is that let's say every cell-free biochemical system up to now that we know, cell-free system that we know up to now, works for a certain amount of time, and works if it's continuously supplied with a flux of matter and energy from outside.
Right.
And then that's the point. So what I would like to work on is to see if I can build, of course, a step by step, some systems that in case of need, or like when they need, they are programmed that so that when they need, they can make use of resources from outside. And this is actually the driving force for them to work and to continue staying out of equilibrium. And this, like, I would like to do for different purposes. So of course, like, from the first one is like to understand how these processes work in life. But on the other side, also, you can imagine, for example, to do materials that have certain properties, and then upon like reaching some condition that could be pH change, could be stress, could be, I mean, it's all to be decided. So they are able to activate a mechanism by which, through metabolism, they can make use again of what is there in the environment and turn it to something that they need. And this is again going towards like a self-sustaining, and also using what is there to perform lifelike action behavior, if you want. Then going back to the first question, it's very hard. The reality is that I'm more expert of the bottom-up approach I described in the presentation. I think there is no right way, is that there are many I think these problems are so big that we should try to all act in complementary ways from different approaches, and try to reach what we want from different angles and perspectives, and ensure that at some point, by integrating again together different approaches, then hopefully we are going to get to something that might be very helpful for all of us.
Wonderful. The tough question and right and good answer. So, but let me summarize. So you believe in the case never dying cell potentially be created
Well,
if you had a completely recyclable material supply and energy supply unlimited.
well, this is like let's say, like very it's a dramatic conclusion, like very strong conclusion. I think that having systems that could be able that are programmed to take advantage of sources from the world outside, and then could also be able to recycle some of the components they have once these components are, for example, proteins are misfolded or are not functional anymore, or are oxidized, or okay, and then as again source of matter and energy to continue to drive their processes. So this, I think, could be way beneficial to build a self-free system that could work for a longer time. Then if it's forever, this is a big statement. I would go step by step, Moon.
Sure. I know I just started the aging project, so I just wonder, I mean, that is possible or not. But it's kind of all kind of the crazy idea, I believe, but I wanted to hear from you. Okay.
Yes.
Now I see the 7 after 11 a.m. Central Time, so let's close and then chat more. Okay, so thank you all for joining and staying today. We'll meet again on August 15, Thursday, the same time, the same Zoom link. We'll have Professor Josh Renard at Northwestern University and Professor Jian Du at University of Hawaii and Manoa, where our first in-person conference will be held on December 9 to 12 this year. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. Thanks. I stop recording. Just give me 1 second.